you can use this one like 1 unit, 1.5 unit, 0.25 unit, because we don't know the milligram gram there. But here we know that this is microgram per ml. So whatever the concentration of unknown solution of methylene blue, that should have the unit microgram per ml. Yes.
So like the last class we also did like we tried to find like observations of all the colors to everything labels for all the colors. What is that?
But wavelength or
That's
as well?
with all the colors because
We had the concentration and we found the wavelength of absorbance. But now we're
If
trying
you
to
have
find
the
out
absorbance
concentration.
data, you can use that one. That is not a problem. Okay, you can use that one. First thing is that for each and every sample, you need to find out the wavelength. Because wavelength is different for each and every sample. You did dilutions there, but you measured the wavelengths, but that wavelength is very nearby. I think so. That is the common result. So whatever, suppose in methylene blue in different solution, if you scan all those dilutions for the wavelength will be similar. Either 668, 667, 669, something like that. It will not change. Wavelength will not change for the sample. If it is a too much variation, that means that is your error. Generally, wavelength will not change per sample. Clear? Any question? So for exam from this part, what you can get that suppose you are given a standard curve there, because in exam you need not to prepare the standard curve. You can prepare the dilutions. So you will be asked to prepare that serial dilutions for that and take the reading. You need not to do the standard curve. Standard curve I will provide you. I will give you one graph with the equation and the R-square value. Okay, that will be provided to you. From there you need to find the concentration of unknown solution. You need to take the reading of unknown solution, put that solution into that equation and find out the concentration of that. Okay, I will not tell you what kind of standard curve is this. Everything will be written there. You need to find out the unit and everything from the graph. Okay. You need not to do the standard curve, but it should be. Suppose if it is microgram per ml, the concentration should be written as concentration microgram per ml or mg per ml, whatever it is. But you need to find out the unit also. And the numbers will be deducted for those. If there is no unit, if didn't you use the equation or something like that, then the numbers will be deducted. Because these are the basic things. When you are learning, I know that you know this thing. How to calculate this. You need not to do too much experiment in exam, but these are the common things. In particularly next day we will be doing the gel, and for gel also we need to prepare a standard curve, and from there we need to find out the molecular weight of a particular band. The same thing, same way we need to prepare that one, but there is some other method. In exam you will be provided a picture of a gel and the total distance of that one, and you need to find out the molecular weight of a particular band. Okay, these are the exam questions. Good or bad? Thanks. And one thing I will just tell you, do the accuracy and precision very well, because in the lab report I didn't find that calculation. What is the precision? Percentage of accuracy and percentage of precision. Not the percentage of precision, percentage of accuracy particularly. Do that, follow that thing very well. Go to the notes and read it again. I didn't find that one in a lot of notebooks. So if the question will be there, to find the accuracy, that means you need to show the percentage of accuracy. Either 99% accurate, 56% accurate, whatever it is. The result will be like that. And 2 or 3 marks will be on that. So if it will be 10 marks, 2 or 3 marks will be the percentage of accuracy. If you do not provide the percentage of accuracy, you'll get 8 or 7.
So what's going to be on the final exam? Is that what you're talking about? Is going to be on the final exam, like after the classes are over, like on December?
Yeah, what you did in the midterm exam, the final exam will be like that.
Oh, so it's going to be all the midterm exams together, kind of.
Yeah, it is together. The whole syllabus.
And when is another midterm next? Is it next?
There is no other midterm exam. We have one quiz, and the quiz will be in this weekend, and that will be for your practice. That is the theory part mainly, and practical part will be in the final exam. And after that, this week is the project due, right? I think the project due in this week, and next week the lab report due. I'll check your lab report. I'll send it to you. So if you want me to check your lab report, you can send me. So during this weekend, I can check and I can make some comments if you want. Okay. Send me the digital copies, and when you will submit the lab report, the full lab report from lab 1 to lab 17, I think, or lab 16. The whole lab report with everything should be there. That is the instruction, manual, the table of contents, page numbering, all those things. Your notebook within the full form. So everything should be there. And send me one digital copy. Okay, the final report. This week. First. Just check the project one. I think project is ready right now. Right? Because you need not to write too many things. No introduction, nothing, just material methods and observation. Result. Those 2 sections. Is it ready yet? Do not make it too late. How many projects are ready? You got almost 2 weeks. Right? Okay. I'll take it on Tuesday. That is the final. And after that I will deduct 10 points. So you'll get another 5 days time. Prepare your report, project report, give it on Tuesday, and that is final. And next Thursday probably the lab report. I think so.
The final, everything.
Because we are done. Next 2 days we will be doing the DNA gel, and after DNA gel we'll have one class for the total review. And another thing is class, I'll go through some of the questions and those parts also. Particularly if you have the problem with graphs. I want everybody that after this semester you are master of graph. Okay, so if you have any kind of problem with the graph, I don't know what you will do, but I want you to be like that you are the master of graphs. So if you have any kind of problem, just let me know with the graphs, so I can go through the graphs again. Yeah, each and every time I need to do it twice. At the beginning of the semester, at the end of the semester, the graphs. Yes.